And welcome to week 11 in environmental science. This week we'll cover environmental health and toxicology, the content from chapter 10 in our textbook. On the first tab of the content page, we have 5 learning objectives. Please focus on these learning objectives as you move through the content in the textbook. This one is pretty closely related to the lab activity that you're going to do. We also have some flashcards or some terms that I want you to know, and I highly recommend that you make flashcards of some of these terms that you're not familiar with. And then finally, there are some tools to help you dive into the material in chapter 10. For example, here's a study table for you to download and fill out. It relates to the information on page 213 and 215. That's a great way to just kind of organize some of the information in the textbook. Here's a similar table, but this one's about toxins that you can download and fill out as you read the content from pages 216 to 218. Here is a video about dose response curves. You're going to be creating a dose response curve in this week's lab. And then the final tab just has one video about risks and assessing versus managing risks. There are 2 discussions in this week's module. One of them is designed to be an extra credit opportunity. So this one involves going to the Environmental Working Group's Skin Deep Guide to Cosmetics, looking up a personal care product that you use in their database, and examining the information that the Environmental Working Group has on that product in terms of its hazard score and also some of the ingredients that are in the product that are of concern, if there are any. So that is due. The initial post about your personal care product and what you learned about it from the database is due Wednesday. And then by Saturday, I'm asking you to follow up and respond to your peers. This is going to be worth a maximum of 5 points if you're using it for extra credit. I say if you're using it for extra credit because our 10-point discussion this week is about a subject that I think is really fascinating. It's about the chemical atrazine and how it impacts different living organisms like frogs and humans, and we're focusing on the work of Tyrone Hayes. So I have Dr. Hayes' webpage, a link to a paper that he wrote in the Proceedings of the National Academy of Sciences, and then a TED Talk, which is a video. So the reason why I have 2 discussions in this week is because Dr. Hayes' research is on vertebrates, specifically frogs, and in his research, he exposes the frogs to the chemical atrazine, which is a common agricultural chemical. This disturbs some students. You know, there's a couple in every class that really don't want to hear about scientific research that's being done on animals. If you do not want to explore this content, then I will allow you to replace your score in this discussion with the score that you get out of 10 points instead of out of 5 in the Environmental Working Group discussion. So you can replace it with the extra credit discussion if you really don't want to explore this material. But I hope you do. I think it's fascinating, and it's a great example of toxicology research. So I'm asking you to answer these questions about atrazine and Dr. Hayes' research. And then I want to hear your personal response to this research. That is due by Wednesday. And then please follow up and respond to at least 2 of your peers by Saturday. So this is worth 10 points. If you really don't want to explore this information, I will replace your score from the extra credit discussion out of 10 for this discussion post, but then you won't be able to earn extra credit for the week. If you do want to do extra credit, then please fill out participate in both of the discussions. This week's lab is about toxicology and is going to require some supplies. So the first part involves calculating an ED, effective dose, 50, for the seeds that you germinated in week 1. You probably have data from week 1 that looks something like this, where the percent germination went down as you increased the salinity. A dose response curve is sort of inverted. It's looking for the effective dose. So we want to know at each of these concentrations how many seeds did not germinate, since that's the effect we thought adding salt would have on the plants. So it's actually going to be the inverse of this graph that you're creating, an inverse of the seed germination graph that you turned in week 2. So to do that, you'll just subtract all these numbers from 100 and graph that. You're also going to draw the ED50 line on your graph. That's going to be the salinity concentration at which 50% of the population was impacted. So that would be right here, around 50%. And you look at the dose at that particular metric. So part 1 involves remaking the graph that you made in week 2 and relating it to the dose response curves that you see in chapter 10, and also drawing the ED50 on the graph. The second part involves you doing what's called a serial dilution. So this is going to require some supplies, some spoons or toothpicks, 1 small container of water, tap water is fine. A set of teaspoons, you're going to need 1/8, 1/4, and 1 teaspoon specifically. A white ice cube tray with 12 wells or 12 white Dixie cups, label tape and a marker, or just a marker if you're using Dixie cups that you can write on, and some food coloring. You're going to follow these instructions to make weaker and weaker and weaker and weaker solutions of the food coloring. As you do that, you're going to fill out a data table that tells us the fraction of molecules, the decimal, the percent of dye molecules, the parts per million, and then you're going to answer this question: is the color visible in the cup? So here's a video that's going to walk you through filling out the data table. So what I'm looking for is a picture of your setup, like all of the cups with the fluids inside, and a data table. And then I'm also looking for the answers to these 7 questions. The first few, 1, 2, 3, relate to the part 1 of the lab, and then the last 4 relate to part 2 of the lab. So please turn this in at the assignment page. I'm looking for a picture of your dose response curve, so that remade graph with the ED50 drawn on the graph, a picture of your serial dilution in either Dixie cups or the ice cube tray, a picture of your completed serial dilution data table, and then the answers to the questions. Like most weeks, we also have a practice quiz. So this is focused on chapter 10 content. This is meant to help you get ready for the graded quiz, which is worth 10 points and due Saturday at the end of the day, so 11:59 p.m. The lab is due at the same time. Please let me know if you have any questions, and have a great week.